And I see yesterday when I was taping as well, the end of parenchyma. I've now deleted stuff. Oh, this isn't my iPad. The other iPad I deleted stuff off a little bit before. Okay, so, right, so we've looked at meristematic tissue, dividing tissue, and we've looked at epidermal tissue, which covers all of the plant organs, and at the tip of roots, like the last couple of, not even near the end, but near, not at the end where the root cap is, but near the end, some of the epidermal cells are modified to form root hairs for more water absorption. But the rest of the root can absorb water, these epidermal cells, but where there's lots of little root hairs, but a bigger surface area. Right, the other two types of modified epidermal cells, if you think of a leaf epidermis, I'll use this. The leaf epidermis can secrete a substance called
waxy cuticle.
A wax, which is, then makes up what we call a cuticle. And the cuticle is not found where?
The root.
On the roots, because that would be against the whole purpose of a root. Okay? And then other modified epidermal cells of which parts of a plant? Would there be guard cells?
Leaves.
Leaves and
green stems.
Green stems, because the outside of the green stem, alright, something like this. There you can see they've drawn a couple of little guard cells, and they've got this, this layer here. It looks sort of opaque. That's the cuticle, and then there's the epidermis. Okay, one layer of cells. Sorry, I need to show you guys as well. Alright, so here. Oh, this is strange. Okay, so there's the outside of the stem, there's a couple of guard cells, here's the cuticle secreted by one, there's the epidermal cells, one layer. Okay, and there could be many layers, depends on the plant. Alright, so that was epidermal, and then we said we need to fill up the plant. And we fill it up with parenchyma or parenchyma. So most of the stem, all the light green here, all of this is parenchyma, and in the middle of the stem, how would that parenchyma differ? Sorry? Guys, your voices must project through a mask.
Wouldn't have chlorophyll.
Who's talking? Okay, wouldn't have chloroplast. Okay, so in the middle we sometimes call it the pith parenchyma. Alright, then underneath or near the edge of the stem, this part here, we call it the cortex, but we'll get to the line diagrams just now. The now I've got to make sure I'm there and there. So we've got all this parenchyma here, all the light green. The middle here is the pith of parenchyma, and the cortex near the edge, and it could contain chloroplast like in a green stem. But in my potato here, are there going to be chloroplast in the cells?
No.
Okay, lots of parenchyma, but no chloroplast. But sometimes, the epidermis or that parenchyma underneath goes green. And I've read an article where they say you mustn't eat green potatoes. You must get rid of the green or not use them because it produces some kind of chemical. Can't remember the name now. Alright, and obviously in my apple, lots of parenchyma and the carrots and butternuts and petals of flowers, everything. Alright, so that was parenchyma. So they were thin-walled cells. I must get a laser. My laser's broken. So they are thin-walled cells. What are the cell walls made of?
Cellulose.
Cellulose. They are living cells, so they've got cytoplasm, nucleus, vacuole, maybe chloroplast, ER, Golgi, all of those organelles that we learned about. What type of section was this again?
Which one?
This one, sorry?
Longitudinal.
Longitudinal section, like cutting a butternut lengthwise, or a cucumber, making those little long frills. And this is known as a cross section or transverse section, cutting across the cells, like when you slice the cucumber. You can see the vacuoles, and you can see the nuclei, but it looks like some of the cells don't have that there. That one doesn't show a vacuole, a nucleus. Because maybe you cut it here, and there's the nucleus. So remember I said it's about perspective. In fact, what I've said now is the part that didn't take at the end of yesterday's lesson. So the people at home can see this now as well. Alright, so now we've got a plant covered with epidermal. It's got growing regions, and it's got parenchyma, but it might be floppy. So what do we need? Parenchyma and sclerenchyma, the hard sounding word. Okay, so the about ground tissue. It's just the word for saying the tissues that form up the bulk of the plant. So now we're looking at the hard sounding word. Oh, look, it's pushed it to the next page. So parenchyma and sclerenchyma. And I remember that I drew, I wonder if it's on here, it must be there. I drew a picture of how this happens. And I showed it to you on, do you remember this?
Oh,
Seriously?
yes.
All right. So let's show you how, if I take a cross-section of parenchyma and make it become parenchyma. Because the cells change according to how that tiny little fruit is growing, and now it needs, like, you know, the core of my apple. Who eats the core? I don't. That hard part. Okay, so there's parenchyma there. It's not nice. It doesn't feel nice when you chew on it. So, in fact, yeah, let me do it the same as I did there. Then you can go look on Google Classroom. So here's the parenchyma cells. All right. And there might be little air spaces between them, like that. And if we draw a cross section, this is the tissue that looks nice, you know, looks pretty under the microscope. And there might be different shapes. You know, they might have that shape as well. Lots of different shapes. All plants are different. Okay, not all, but there is variation. So there's a cross section and a longitudinal section of parenchyma, and there's a vacuole, cytoplasm, nucleus, and all the other organelles. So in order for this to become parenchyma, what do I add? More cellulose. C for cellulose. Okay, so I'm going to do them in different colors though. So it's almost like the cell walls get thickened, but more so at their corners. Let's draw another one there. So if this cell wall is going to thicken with extra cellulose. I never understood this at school. Didn't make absolutely any sense to me. But if you think about it, it's pushing out the air spaces. They're disappearing now. So it looks like the cells get corners, like they become almost like little hexagons or whatever.
Where do they get the more cellulose from?
They make it.
So parenchyma turns into
Yeah, so remember they're making glucose, then they can make fructose, they can join them to make sucrose, like in cane sugar. Some glucose can change to amino acids, or some glucoses can join to become cellulose. Do you remember polysaccharides? Glucose becomes cellulose. Glucose can be stored as another polysaccharide, starch. So here the glucose molecules are joined to make more cellulose at the corners. And that's why it has that very distinctive shape when you look at it under the microscope. Okay, and then obviously here, I'm not going to show everything, but the cell walls do become thicker, and the air spaces are obliterated. They're squashed out. All right, and that's what it looks like. Now, they are still living cells. So because they've only got cellulose, it's permeable. Things can still move through the cells, so they still have cytoplasm, vacuoles, nuclei, and all the other organelles. But they can perform a supportive function as well. So, celery, I will set up the microscopes. I've got a date for next week. But if I pull those bits, all right, so celery has got parenchyma under the epidermis. So if this, I'm just trying to think how this is looking now. Oops. Okay, so that's my celery. Cross section through a stick of celery. There's the epidermis, and it's quite hard to find the parenchyma, but it's just under the epidermis there. So if you had to draw it from the side, there you'd see lots. If you looked at the cells from the side, you'd see lots of parenchyma cells like that, but from the top, you'd see that. And these are the stringy bits.
Oh, is that the...
That's why it's tougher when you eat it. Okay. All right, so I'm going to use this to actually try and find, it's quite hard because you've cut a really thin section. And then this little area here, it shows up as a grey colour, and then there's lots of parenchyma here with chloroplasts, and then you can see the epidermis as well. Oh, and then you would see the veins, the xylem and phloem as well. But I'll show you when I set up the microscopes. All right, are we happy with that? Okay, so let's have a look here now. So parenchyma, oh, here it is, and I wanted this. So it's broken. Okay, improvise. Okay, so there's a parenchyma cell, more or less. More or less hexagonal in shape because the cell walls have become more thickened at the corners, but they're still living cells. Whoops, I need to see as well. Okay, thickened at the corners there. And that's my little broken model. I think that's from transporting it home and fix that up. All right, so what do we have here? They are living cells with cytoplasm. And the cell walls are unevenly thickened at their corners, not really, but corners with extracellulose, and that's why they serve as strengthening. And it could be, near the veins. So the veins, if I remember correctly, are a little bit inwards. And even in a leaf, there could be parenchyma. So let me get that model. Okay, we're going to be looking inside the leaf. Here's the mid vein. Okay, there's parenchyma. If you get a chance, come and have a look at the model. You can see that they've tried to model those extra thickenings here. So that's parenchyma. In other plants, in other leaves, it might only be at the top and the bottom, or none, because this one's also got sclerenchyma, and xylem and phloem. Just depends. Leaves are different species. Oh gosh, I'm not showing them. Okay, so there's parenchyma around the middle vein, and there's sclerenchyma. But in a stem, all right, cross section of a stem. This is really hard. I'm not used to doing this. Okay, there's the parenchyma below the epidermis. They've just colored it in dark green just to make it stand out on the model. And then you can see the cells from the side. There they are. And not all stems will have them. Sometimes they might just have patches of it. Sometimes rings. It just depends. Around the apple core, there's a bit of parenchyma there as well. Okay, guys, we happy?
Yes, sir.
Great. What else is there? Oh, the cells are tightly packed because those cells, there might still be a few air spaces, but they've been squashed out. And the chloroplasts as well, if they're near the edge of stems, or even in the middle of the leaf, maybe they still have chloroplasts. Just below the epidermis, mostly below the stem, or around the vein of a leaf, the middle vein of a leaf. Okay, so that's parenchyma. Right. Now we look at the next strengthening tissue, which is sclerenchyma. So you now have to imagine a rectangular prism or an oblong-shaped cell becoming that. So what gets added to sclerenchyma cell walls? Lignin. So lignin gets added to the cellulose, and sclerenchyma cells are long, thin, and pointed. We say they are tapered at their ends. Tapered. Okay? So now what I want you to imagine, okay, I'm going to draw on here. Pretend that I'm drawing the cell from the outside. So there, that cell is covered in cellulose. Okay? Think of me coloring a butternut, I don't know. Coloring in the cellulose all around. But now I'm going to add lignin. But not all over the place. I'm going to leave little areas where there is no lignin. This pen's running out. You see what I've done? I've left little openings of cellulose. Can you see what I've done here? There. I've poked little holes in this very old roller towel thing. These little holes, are they holes? No. They're called pits. So that little bit, wrong color, where there's still a little bit of cellulose in the wall, it's not a hole, it's a pit of cellulose. And the rest of the cell wall has been strengthened with lignin. And lignin is a tough waterproof substance. And it helps to make wood. So wood is mainly made up of cells that are strengthened. The cell walls are strengthened with lignin. Okay, so can you see how a parenchyma cell can become a sclerenchyma cell? All right, extracellulose gets added. Is this all in the way? I need more space here somewhere. Okay, so that is now a sclerenchyma cell with pits of cellulose, because remember cellulose is permeable. So it's easy for water to move through these cells. They don't form a barrier because lignin is impermeable to water. Water can still pass through the cells, but they're there. They're like the columns, the big concrete columns in a building. Have you guys ever watched on DSTV where they show you how they make these buildings? It's amazing. I watched a couple of episodes in the holidays. It's quite amazing, or how they make a ship, and then they've got these huge columns of metal or concrete in a building. All right, so that's what these cells do. What's inside? What was inside before it became a sclerenchyma cell?
Cytoplasm.
There was cytoplasm. But the minute there is lignin, what happens inside?
It dies.
The cytoplasm dies. So they are hollow supporting cells. Okay? And look what it says. The cell walls are thickened with lignin to strengthen them. I know you can see on your notes. Keep it at home as well. All right? The cells are dead and hollow. Now, I haven't explained the fibers. They're long tapering cells with pointed ends. So what I'm holding are sclerenchyma fibers. Yes?
Why do the cells have cellulose bits if they're dead?
So that water, okay, where's blue? So that, there might be parenchyma over here, and there's more parenchyma. So water can pass through. They don't form a barrier. Because imagine if there's a whole ring of sclerenchyma fibers, and there's parenchyma in the middle and there, now water can't go across. Okay, so they don't form a barrier. All right. Okay, so fibers, these ones serve as strengthening tissue, like in plant stems and around the veins of a leaf or a stem. So let me show you on the model. Remember? As long as you just, oh gosh, here's my little leaves. Somewhere, yeah. Okay, remember you're looking inside the leaf, hey? Top to bottom. So there are sclerenchyma. Okay, there are sclerenchyma fibers. There's a cross section. You need to come and have a look at this model. And then if I look from the side, can you maybe see that they're long and thin? There they are. And they haven't at the top. There they are. They're arranged next to each other there, like that. Okay. Right, but now you get another type of sclerenchyma cell, which looks totally different. And what are they called? Sclerids. Okay, so let's get rid of this. Okay, and they probably developed from parenchyma cells. And this is really nice to look under the microscope. And that's why I need to go buy pairs. And when I buy the pair, I bruise it. So I need to get them today so that by next week they're ready. So where the bruised part, under the skin, that's where you're going to find lots of sclerids. And they're really pretty under the microscope. Okay, that's a botanist's thrill, okay? When I find those, then I'm happy. All right. So what happens here is the parenchyma cells, remember they have got a vacuole nucleus cytoplasm. So what gets added to the cell wall to make them sclerenchyma? Lignin. And it gets added in layers. So one, okay. So as the cell wall thickens, it forms layers as it grows inwards. So there's all this lignin. It becomes like a little, no, not really, like a little concrete ball, not really though. But it can't be impenetrable to water. So little pits of cellulose, little channels of cellulose are still present in that lignified wall, so that water can pass through, through that channel and then out the other side and maybe into another parenchyma cell. All right, so do you understand that process? Okay, lignin gets added in layers. And that's why they look like that under the, there it is. That's exactly what they look like under the microscope. They're sometimes called stone cells. And you find them in clusters, and it's really pretty under the microscope. All right. And from the outside, I mean, if you look here, show the people here. Can you see these just little holes that have been poked through? But they would be little channels of cellulose, so that water can pass through. Okay? And I've kind of drawn some lines there to show the layers of lignin. Can we see that from the back? Sort of. Can you see? Okay, guys, are you happy?
Mm-hmm.
All right, so here, sclerids, maybe I shouldn't say short. They're more rounded. They're pitted. So they've got these cellulose pits. That's a pit. Yes.
Why are they only found in the seed coats, mudshells, and in tails?
It's just the way those structures are formed, those fruits and veg and whatever. It's just the way it is. They just provide a little bit of support. Who eats pears? And you eat into the pear and there's that grittiness.
Mm-hmm.
That's what it is. I don't, and guavas. Who eats guavas?
Yeah, I love guavas.
Okay, and the shells of nuts. What else did I say in there? Oh, it's here. Seed coats, the seed coat. So hard seeds or the shells of nuts. And nuts is a fruit. So if you have, I've actually got macadamia nuts in my car that I picked up the other day. I should bring it in. There's the seed, and it's got this hard, who eats walnuts or things like that? So that hard fruit wall, it's actually, a nut is actually a fruit. So that wall could have lots of sclerids in it to make it really, really hard and you know, lots of lignified cells. But then you've got the seed in the middle, and there could be cells, parenchyma cells here that are storing food. And there you've got that little embryo, which is dividing by mitosis. So there's lots of meristematic tissue, and then it becomes the root and the leaf.
Ma'am, is the pit for the sclerids in that line? Oh,
Yeah.
okay.
So these little channels of cellulose passing through that extra thickened lignified wall. And now you can see that there's a little space in the middle. And what do we always call a space, like if you think of mitochondria?
Lumen.
Yes. Okay, so remember the space in the middle here, we called it a lumen. So that little space in the middle, you can call it a lumen. I didn't really put it over here. But there's no, it's dead now. So you would see lots of them in little clumps, lots of them. And they overlap. Okay, so that's what we're going to have a look at. And then you would see all the parenchyma, the pear tissue around it. That's what we're going to look at. All right, have I said everything? Yes. Okay. What time does this end? Oh,
Yeah, double.
yeah, I know.
How do you spell lumen?
Lumen. L-U-M-E-N. Okay, so now we've got a plant that's standing up more. It's got some strength. But plants, what do they also need to do? They need to transport things around. Because they're absorbing things through their roots. It needs to go up into all the cells, and they absorb and making things in their leaves. And what else? They're also absorbing stuff through their leaves. So we need to figure all that out. Okay. So the vascular tissue. Vascular tissues, xylem and phloem. Xylem transports. Right. So who has added fertilizer to their lawns or their gardens now?
Mm-hmm.
You're waiting for the rain. You want those mineral salts to dissolve, and then they get absorbed by plants, and then they can grow better. Nitrates. Nitrates are a good source of growth. Phosphorus, calcium, all of those things help the plants and the roots to develop. So if you plant a new plant, you put bone meal in the hole. Yes? No. Guys, you need to go garden. So calcium, bone meal, and phosphorus in that, it helps when you plant a new tree or something, the roots grow better. And then they can absorb those nutrients. You can add, the other day I ran out and I added bounce back, that chicken poo pellets. And then it dissolves and it becomes the nutrients. like little granules, natural, or you can go buy fertilizers. Okay. Sorry?
Or compost.
Yeah, compost is broken down plant organic material. So it becomes minerals and nutrients and, yes, well not nutrients, minerals and other things. I don't like to use nutrients. I like nutrients for amino acids, glucose, etc. Can we have a break, please?
Yes.
Okay, so when the bowl starts, we're out of break, we can do xylem and phloem. Come and have, if you want to, come and have a look at the models. I'll put them back there. On the side here. You can come and have a look over here. You can come one by one if you want to. Come see if you can recognize the parts. Who wants to? Nobody?
I'm coming.
Nobody? There's another one next door. Yes. Now you're going to listen. Come on. Come here. Let's fix the model.
Nothing about you, don't worry.
Securing your future and ready. Don't worry.
Huh?
I'm securing your future. Don't worry.
Can we tell them? Should we tell them?
Tell us.
We just want people to like play games to help us remind like people who.
Yeah.
So nobody wants to have a look at these?
I do. Me too.
Go see if you can see the tissues. But obviously stay apart, guys. Stay apart. Nobody else.
Yes. I need to buy.
You'll just stare at me. I just see you. Huh?
Yeah, I think so.
Wait, Mom. So this one is securing here.
Colin.
Colin.
See? Yes. Red silent. There's a bit of movement tissue there. They seem to be colour and yellow. Vascular cameras. Well, I don't put that in, but I'll spongy.
What's that? The muscle one?
These are for middle of the knee.
Oh.
All the spongy and the cameras. Oh, we have today. Okay.
Does your boy actually
Yeah.
I think I'm going to switch